Hello, I'm Krisha, a field application scientist at Akadeum, and today we're taking a closer look at our human T-cell Leukopak isolation kit, GMP grade. This kit was specifically designed for clinical and commercial manufacturing workflows, where maintaining cell quality, workflow flexibility, and regulatory compliance are critical. Why choose Akadeum? The answer lies in our unique buoyancy-based microbubble technology. Microbubbles bind to the unwanted cells and float them to the top of the bag, allowing T cells to settle near the bottom, where they can be transferred manually or using instrumentation already available in your lab. No specialized instrument is necessary. This allows for fewer steps, minimal cell handling time, and a scalable cell isolation that integrates directly into closed system, GMP-compliant workflows. Included in this kit are our streptavidin microbubbles, our negative selection antibody cocktail, and buffer. Standard cell processing bags, an end-over-end rotator, and a tube welder are also needed for this workflow, but not included in the kit. Post-isolation, T cells are ready for cell culture or other downstream assays, including single-cell sequencing, flow cytometry, or other functional studies. Akadeum's GMP-grade human T-cell Leukopak isolation kit offers a GMP-compliant, scalable, high-performing 60-minute workflow that maintains the health of your starting sample. If you would like to learn more or request a demo, visit us at akadeum.com.